Okay, so let's look at what we need to know, just the kind of major topics here. I'm going to go ahead and begin with chapter 23. Make sure you're able to classify your structures into, upper and lower versus conducting and respiratory zones. So know what belongs where, because that's important to know. Uh, and so that's that. About your epithelium here, about the mucosa, make sure that you are able to tell me that the epithelium does change and gets progressively thinner as we go from the upper respiratory tract towards the lower. Know your exceptions. So where do we find the non-keratinized stratified squamous epithelium? So know those areas and why. Okay, understand why there are regions that have this non-keratinized stratified squamous epithelium? And the answer is because this is also shared with the digestive system. And so non-keratinized stratified squamous is able to withstand abrasions from the food passing through. You don't need
Sorry,
to.
Prof. What? Really quickly, is that your on page, or sorry, figure 23.2? Should we have that down pat? What you're talking about?
Yes, I don't think I'm going to go into like where are we going to find simple ciliated columnar versus cuboidal, things like that. Just for that, the progressively thinner epithelium, the yellow box, just know that it gets progressively thinner. But for the exceptions, yes, know the exceptions. Does that help?
Yes, that helps.
Okay. So going on here, the next section talks about the upper respiratory tract, the nose, just structure, make sure that you are able to label things. And also as you're reading this, understand like how these structures work together. So like, for example, those conchae, they help create the turbulence and separate the nasal cavity into sections. So, so that's that. You don't really have to pay attention to paranasal sinuses because I don't really like that figure. It's not very good. Yes, Hazel.
The upper respiratory tract. I understood that the conchae are the bony parts, and the meatus are the canals. But I still don't understand the difference between the conae and the conchae.
Oh, choana is
Coena.
it's just a space that it's kind of like a border between the nasal cavity and the pharynx. So it's that narrowed part, the posterior narrow part of the nasal cavity. Yeah.
That's separate from the meatuses.
Yes, it's like the meatus kind of join into that.
Okay, I think I'll look at it again. Thank you.
Right. And yeah, what else here? So yeah, because if you look at that picture there, it's a bracket, so it's just that kind of that space, that narrowed space at the end. Hold on. Seems like Jose is being kicked off from this. All right, and I won't make you label the cadaver here. So just know the figures. Pharynx, I think, is pretty straightforward. Know that this is the first place where we see the exceptions in the oropharynx and laryngopharynx for the epithelium. Larynx, be able to label it, and that's that. Make sure that you know the sound production, the things that affect the sound production, and we talked about that in our first class. So pitch, range, and loudness, how are they affected? And that is that. Understand that it's the arytenoid cartilages that rotate, and they are going to change the distance between the ligaments as well as the tension on the ligaments, and that's what's going to modify the voice or the sound that is being produced. For the bronchial tree, know the basic structures. So know that your trachea is it's kind of like the trunk, and then you have branching going on all the way until you get to alveoli. Be able to tell me we have the main or primary bronchi, then we have lobar or secondary bronchi. Okay, so those terms go together. And then we have tertiary or segmental bronchi, and kind of how they correspond to the division of the lungs. And then after the tertiary bronchi is smaller and smaller bronchi, be aware that the difference between bronchioles and bronchi is in the cartilage. Yes. Hazel, sorry.
Oh, I don't have any. Oh, I guess my hand is still
Oh,
raised.
yeah, I just noticed that. Okay, so that's that for the bronchi. We have figure 2310 shows you the whole pathway of the bronchial tree there, so you can see the cartilage is what differentiates between the bronchi and the bronchioles, and bronchoconstriction, bronchodilation is shown there as well. When you get to figure 2311, and here, no. This is very tricky to label. So if you see this one, notice which one is the bronchiole and which one is the artery and which one is the vein. A lot of times I have students mistakenly label the bronchiole as the blood vessel. That's not a blood vessel. Okay, it looks red and it's right next to the blue, which is a blood vessel, which is the artery, but it's not. That's a bronchiole there. Okay, so pay attention to the labeling here. And
Are you talking about 2316?
2311. Bronchioles and alveoli. I'm looking at that's upside down, but it's this one. So in there, make sure that you're labeling those vessels right. And then the alveolar ducts and respiratory bronchiole and terminal bronchiole, those are important. So I think whenever you're labeling this, I think it's easier to go backwards. So you have alveoli, then alveolar ducts, then before that is respiratory bronchiole, and then before that is terminal bronchiole. So make sure you know that. Terminal bronchiole is the last part of the conducting zone, and respiratory bronchiole is the first part of the respiratory zone. Figure 2312, when you look at that, again, make sure that you're able to label these cells, but also know what the function of these cells is. So we have the alveolar type 1 and alveolar type 2. Okay, Jose says that he can't hear anything. Sometimes that happens to me. You might want to like restart Zoom or restart your computer. My Mac has been doing that lately, because I think Hazel can hear me, right?
I can hear you.
Yeah, okay. So Jose, if you want to like restart and then come back, that might help. All right, so 2312, again, alveolar type 1 and 2 cells, know the difference between them, know the parts of respiratory membrane. How does the gas move from alveoli into the blood and the other way around? Remember that from alveoli into the blood, you have oxygen moving into the blood, and then carbon dioxide is going the other direction. So be able to kind of match these events. Okay, so oxygen will from alveoli pass the respiratory membrane, it will go into the blood, and then blood carries it to the rest of your body, will be exchanged the other way. And then carbon dioxide is always going to be doing the opposite of what oxygen is doing. So that is that. Table 23.1 kind of nicely summarizes everything for you there. So summarize basically everything that has been said up to this point, at least for the lower respiratory tract. For the lungs, make sure that you know lungs are covered by the pleural membranes. We have the parietal and visceral membranes, so understand that. Between these two membranes is a potential space, and okay, diseases and abnormalities you need to know. Not really. I think if you read it, it might help connect things for you, but I'm not going to specifically ask you anything about pneumonia or something like that. But I think reading it is just kind of like an enrichment and helps you put things together. Okay, so pleural membranes, they're going to, the visceral layer is attached to the lung, so it's the outer portion of the lung. The parietal layer is attached to the thoracic cavity. And so as you are working on expanding your thoracic cavity, the parietal layer or parietal pleura is going to expand with it, and that is going to pull the visceral pleura, which helps stretch the lungs. So there's kind of like that change in pressure whenever you look in the chapter later. Make sure you're able to label lungs, the lobes, the fissures, and the vessels, hilum, things like that. So for figure 2314, make sure that you are able to label that. Figure 2315, I'm not going to ask you to label segments, okay? So you don't have to label segments, just the lobes, and know which one is left and which one is right lung. And which one has 2 lobes, which one has 3 lobes, right? So that's that. Figure 2316, you have the blood circulation here shown as well. So this is the pulmonary circulation. Remember, in pulmonary circulation, the arteries are blue and the veins are red, because in pulmonary circulation, this is the movement of blood between the heart and the lungs. So from the heart, deoxygenated blood leaves the heart through an artery into the lungs, and then oxygenated blood will come from the lungs to the heart. So the colors of the veins and arteries are switched for pulmonary circulation. The pleural membranes I kind of already mentioned briefly here, and that goes with how the lungs remain inflated. For the process of respiration, make sure you know that there are 4 steps there. You have your pulmonary ventilation, and that's the exchange of air between the environment and your lungs. So it's moving the air into your lungs. And then you have gas exchange. This is an alveolar gas exchange. And alveolar gas exchange is also known as external respiration. And then the gas has to be transported once. So for tracing pathway of oxygen, okay, now the oxygen would move into the blood and it has to be transported. Then there will be systemic gas exchange here, and that's going to move oxygen from the blood to the cells, so the cells can use it. And then carbon dioxide will move the opposite way from the cells into the blood. And so you can see that the gases always move in opposite directions there. So that's that. Pulmonary ventilation. Then it goes into those individual steps. For the muscles, just know which muscles go with which type of respiration or which type of breathing. You do not have to, I'm not going to make you label them, and you do not have to like tell me about them. Just strictly be able to match the bold stuff in that figure 2319. Okay, just match the bold stuff. Know which muscles go with what. By the way, like if you are seeing muscles of quiet breathing are diaphragm and external intercostals, and you don't really see them listed on muscles of forced inspiration, they're still going to be used there. These are just additional muscles that you're using for that forced inspiration, because in the forced inspiration or deep inspiration, you have to expand thoracic cavity even more than you would at rest. So those are additional muscles there. For the pressure, yeah.
All right, so we would do well, that table at the bottom, we would do well to memorize that? The muscles of breathing table?
Yes, but just the bold stuff.
Yes. Okay, thank you.
So you might want to like rewrite the table where you just get the 3 types of breathing and then the muscles.
They're all bold.
Yeah. All right, the Boyle's law. Know briefly what Boyle's law is. I want you to know how the pressures change, but not necessarily like you don't have to memorize the numbers. If you do see numbers, then you will be able to infer like changes, is it increasing or is it decreasing? Like there won't be random numbers there for you. But I want you to be aware, like how does the pressure change when your lungs would expand? The pressure drops in your lungs initially, and that's what causes the air to move into your lungs. So just be aware of like increases versus decreases kind of stuff. I would recommend you memorize the 760 as the standard pressure. That might be useful later on in other classes as well, because we use the 760 millimeters of mercury as a standard pressure. So, and that's pressure at like standard conditions. It's like, yeah, so. So our atmospheric pressure is somewhere close to that, but we're using 760 for illustration purposes, okay? The nervous control of the breathing. I think this figure is really, really helpful. Armando on YouTube also has a really excellent explanation of this if you're following him on YouTube. So you might want to check him out, but I summarized that for you on the I think it's on the Nearpod, so you can look at that, be able to tell me, you know, what regulates the respiratory system. But I really like that our figure 2323, because you can kind of see what's coming from where. Okay, that's a very long 2 pages after you flip from that figure. So that's why, you know, if you just watch the lecture or watch the Nearpod, and it's discussed there, and it might be shorter than reading those 2 pages. For the rest of this, like I'm on page 932 in the hard copy of the textbook, like airflow, pressure gradients, resistance, know your terminology, know what is what, know the definitions from that section there. Pulmonary and alveolar ventilation. So we said pulmonary ventilation, it refers to breathing. It can also be the amount of air, and it's the amount of air that's exchanged between the atmosphere and your lungs in a minute. Then know what's anatomic dead space, what is the physiologic dead space. Be aware that these two in a healthy individual with healthy lung are the same, but somebody that doesn't have healthy lungs or have some kind of obstruction or loss of alveoli, their physiologic dead space would be higher, meaning that more air that enters the respiratory system is unable to go through gas exchange. So that's that. Anatomic dead space is simply air that's stuck in the conducting zone. So it's not going through gas exchange. For the volumes and capacities, you don't have to memorize all those numbers there. I would like you to be aware kind of like what is what here, but know your tidal volume. Tidal volume for both males and females is 500 milliliters. And then the total lung capacity, which for males is 6 liters and for females is 4.2 liters. So those numbers, do you know? I'm not going to ask you any other numbers. If you by any chance see any other numbers on the exam, let me know. The next section, respiration, alveolar systemic gas exchange. This again, if you look at the figure 2325, I really like to work with pictures and talk about stuff when you're looking at the images. So if you remember what we talked about the first time when we talked about this, you know, maybe re-listen to that part and just see how the gases change in partial pressure and therefore their concentration in both cases here. So when you inhale, now your lungs are going to bring a lot of oxygen into your lungs. And the concentration of oxygen and therefore the partial pressure of oxygen in your lungs will be higher than what it is in the deoxygenated blood. And so by the simply by concentration gradient, the oxygen is going to move into the blood. And then for carbon dioxide, it's reversed. Carbon dioxide is higher in the blood, in the deoxygenated blood, than in the lungs that now have this fresh air in there. So carbon dioxide would diffuse out. So concentration and partial pressure are directly related. The more of the gas you have, the higher its partial pressure is. Okay, so you can think about those numbers, not just in terms of pressure, but you can also see, okay, I have a lot of oxygen in the alveolus and I have less oxygen in the blood. Okay, and you can think about it in terms of concentration as well. Know what's a ventilation and perfusion coupling, and just kind of like basics of how that works. Basically, this is how lungs and blood flow both kind of adjust to maximize gas exchange. That's that in a very brief description. For gas transport, know your terminology here, know how oxygen is transported through your blood, and this is mostly through hemoglobin, but very small amount of oxygen is dissolved in the plasma. And that I'm not talking about the O in the H2O, I'm talking about O among H2O molecules, okay, like O2 with H2O molecules. But oxygen is not very soluble, so only about 2%, very small amount is dissolved. Most of oxygen travels on hemoglobin. Carbon dioxide is a little different. This one does travel through hemoglobin also. It also travels as dissolved in the plasma, and it's more soluble. But the most of carbon dioxide travels in the form of bicarbonate. So know that. We will come back to bicarbonate later. Actually, you see a little bit of bicarbonate formation in digestive system also, and we'll talk about it in urinary system as well, because bicarbonate is a very important buffering system. Oxygen hemoglobin saturation curve. Watch Armando's video. It is posted on Blackboard for you, and he draws it really nicely. So, but know like the effects of different things on the curve, and what does it mean shift to the left versus shift to the right. And so that's that for the oxygen saturation curve. And then breathing rate and homeostasis. Be able to tell me what is you know what, I just realized I was talking to Jose and he said he can't hear me. And wow. Anyway, know your the terms hyperventilation, hypoventilation, and then briefly be able to describe the hyperpnea, which is associated with exercise. Know the terms that go together. So in hyperventilation, you have the hypocapnia and respiratory alkalosis, and hypoventilation is respiratory acidosis, and then the hypercapnia. Let me write a note to Jose. So, okay. Anyway, so, and that's that concludes chapter 23. Okay, now let's look at 26. So we're just talking about 26, the first part on Monday. So hopefully you were, okay, yeah, you got my message now. So hopefully you were okay with that, but make sure that you are familiar with the structure of the GI tract. Know the classification, as always, as with respiratory system, you know, upper and lower respiratory system. Here we're going to be talking about the upper and lower GI tract. Okay, duodenum is part of the upper GI tract, even though it's part of the small intestine and it's discussed under the section with the lower GI tract, it technically belongs into upper. Be able to tell me which ones are accessory and which one accessory organs, and which organs or parts are parts of actual digestive system or GI tract, not digestive system. Be able to describe those different processes. They're at the beginning of the chapter. The control of the digestion. So we have know what enteric nervous system is and why it has a specific name. And then autonomic nervous system. Okay, let's see. That would be a quick restart. Can you hear me now? I don't know if he can hear me. Oh, no, wait. It says he's not here. Okay. All right. The long reflexes, short reflexes, what is the difference between them? Be able to tell me that. And then hormonal control. This would be a good place to write down all 4 hormones that are involved in the digestive system. And these are gastrin, cholecystokinin, secretin, and motilin. And then know what these 4 do. And they're discussed wherever, in the part where they are being released from. For the membranes here, be able to tell me which ones are which. We only have, we don't have a picture of these, so you only have a cadaver, but know what these membranes are associated with. The greater omentum, lesser omentum, what else? We have falciform ligament, the round ligament. You can see when we talk about the liver, those ones that are associated with the liver, you can see in that liver section. And oral cavity, labeling, so know what is what there. That's that for oral cavity. For your teeth, make sure you are able to tell me the structure of the tooth, individual tooth, and then know the teeth, which ones are which. Okay, there is a system to it. So you start with your incisors and then work your way towards the back. There are more teeth for the permanent, on the permanent teeth side than the deciduous teeth. You do not have to memorize the times. Figure 26.4, hold on. Figure 26.4, just make sure that you're able to label the things, okay, and know what is what there. For the teeth, you do not have to tell me the months or the years whenever they're coming in or when they're falling out. Just know what are the teeth called, okay? For pharynx and esophagus, there's not a whole lot that we say about pharynx and esophagus there. Esophagus, make sure that you know the differences in the muscle, that you start with the skeletal muscle. In the upper region, then it's 50-50 skeletal and smooth, and then there's smooth muscle at the bottom part of the esophagus. And esophagus passes through what we call esophageal hiatus, and that's the opening in the diaphragm. Phases of swallowing, first one is voluntary, that's moving bolus towards the oropharynx. When the bolus enters oropharynx, that's when the involuntary pharyngeal phase kicks in, and that contracts muscles and pushes the bolus into the esophagus, and that's your esophageal phase. So those are the 3 phases of deglutition or swallowing. And then for stomach, label the stomach, and then be aware of the sphincter and how that's the place where the epithelium changes very, very suddenly. So you have esophagus, you have the non-keratinized stratified squamous epithelium, but stomach is going to have columnar epithelium. So simple columnar epithelium, it's a very, very sudden, sharp change. Know the main regions of the stomach, of course. And then for the cell, like when we're looking at the stomach wall, figure 2610, know what these cells are doing, okay? Be able to match the secretion with the type of cell. Know which cells are found in the stomach, because sometimes students confuse some cells that are in the stomach with the cells that are in the small intestine. So in the stomach, you have 5 different types of cells. In the small intestine, you have what, like 4? It's not 5, or is it just 3? 4. Okay, and they're not the same. So match the secretions to the cells and know which cells are found in stomach, and then which cells are found in the small intestine, and also what those do. So that's that. And for motility of the stomach, gastric mixing, that's we know that. So make sure that if you just read through that part, once or twice, you'll be fine with that. Regulation of the digestive processes. Know that the contractions of the stomach occur at the same interval all the time, so about 4 times a minute. And then when you're increasing the mixing of the stomach, you're not increasing the frequency of the contractions, you're increasing the strength of the contractions. So look at your cephalic phase, gastric phase, and intestinal phase in figure 2614, and be familiar with what stimulates each phase and what is the result of that phase. So your cephalic phase and gastric phase are going to like promote digestive system things, processes, and then the intestinal phase is going to not inhibit, but decrease its secretions and contractions, because this is the intestinal phase kicks in after as the chyme exits the stomach. So the contractions of the stomach then decrease. Okay, so those are those 3 phases. For your small intestine here, I've already mentioned duodenum is part of the upper GI tract. Here we're gonna focus on histology here. So know that the surface area of the small intestine is very high, and it's established by numerous like different folds and structures. These are from the largest to the smallest. Plicae, those are your large folds. Plicae are aligned by villi, so that would be like smaller than the plicae. And then each villus is lined by microvilli. So those cells that make up the villus are lined by microvilli. You can't really see them very much. It's the darker kind of line on the outside of that villus. When you look at, if you were to look at the microscope or under the microscope at this, you would not really see the microvilli individually. You would just kind of see this fuzzy edge on there. And so we also know villi as the brush border. So when you hear the brush border enzymes, you're talking about enzymes that are active at this area in the brush border in the microvilli. So if you were to arrange the structures that contribute to increase in surface area of the small intestine from the largest to the smallest, you would say plicae is the largest, then villi, then microvilli. Okay. Sorry, Hazel, I just saw your question. And hormones released in each phase for the cephalic, gastric phase, and intestinal phase. Yes. Yes, know the hormones that are going with that as well. Okay. Motility in the small intestine, this is going to be segmentation or mixing. That's just back and forth, not moving things down the line. But as mixing is happening, the nutrients are being, and water also, are being absorbed into the blood. So the contents of the small intestine decrease. And this is going to decrease stretch of the small intestine. And this decrease in distension or decrease in stretch as you're absorbing things into your blood is going to stimulate peristaltic contractions. So if you have not seen the Nearpod yet, that's where I talk about it, how these peristaltic contractions or peristaltic waves happen. So make sure you look at that. Accessory structures, here we're talking about gallbladder, liver, and pancreas. Gallbladder and liver are going to release bile salts, or bile, which has bile salts. And bile is going to help you with digestion of lipids. Bile helps with mechanical digestion of lipids. Bile is not responsible for chemical breakdown. It only mechanically separates large lipid droplets into smaller ones. When you look at figure 2618, what's important here is also to realize what the ducts are doing here. So make sure that you're able to label ducts. So on that figure 2618, imagine the liver right there where it says right and left hepatic ducts. Okay, that's where the liver would be. So the bile, as it's produced by the liver, is collected by small ducts that ultimately merge into left and right hepatic duct. And that's one for each lobe, because the liver, a human liver, has 2 lobes. And then these together merge into common bile duct. Then there's another duct that connects to the common, did I say common bile duct? Hepatic. That's what I meant. Okay, let me start again. Okay, so you've got your left and right hepatic ducts. They merge together into common hepatic duct. And then you have a cystic duct that connects to the common hepatic duct. And then these two, the cystic duct and the common hepatic duct, when they merge together, the resulting duct is going to be called common bile duct. So go over this several times because it can get tricky. And then this common bile duct will empty into the duodenum. And right before it connects to the duodenum, it will fuse with the pancreatic duct. And when they fuse together, they create this kind of enlarged area, and that's known as the hepatopancreatic ampulla. And the opening into the small intestine is your duodenal papilla. Okay, so know your ducts, make sure that you're able to label them there. Gross anatomy of the liver, make sure you're able to label. Again, there you can see some of those ducts, well, a couple of them at least, on that picture there. Be able to label the lobe. Know that liver is your largest visceral organ. I think you might already know that from AP1, I don't know. And then the histology of the liver, be aware what's going on there with the cells, that they're arranged in a hexagonal lobule or hexagonal lobules. And at the corner of each lobule, you have that portal triad. So know what the portal triad is made out of there, and just kind of general pathway of blood and where this blood is coming from here. So liver is going to receive blood from two places. It's going to receive blood from the hepatic artery, and that's coming from the systemic circulation. Okay, this is oxygenated blood, and it brings nutrients and oxygen to the liver and keeps the liver alive. But there's another artery that is going to bring blood to the liver, and this is the hepatic portal. Not the artery, hepatic portal vein. And this one is going to bring blood that's different than in your regular veins. This blood is going to be rich in nutrients, but it's oxygen poor. Okay, so the hepatic portal vein is bringing blood from your digestive system, okay, from your GI tract. This is after it picked up all the nutrients, but also delivered oxygen to the organs of the GI tract. So it's oxygen poor, nutrient rich in the hepatic portal vein. And when this blood flows through the liver will process various molecules, metabolize various molecules, purify the blood, eliminate any pathogens that might be found there. And that's what those reticular endothelial cells, or also known as the Kupffer cells, are for. And then we can send the blood into the regular circulation. You can see a picture of liver that is going through cirrhosis. And then you can, on the next page, you can see what gallstones look like. Yep, they're real stones. So if you're interested, you can look at that. I'm not going to ask you questions about that, but again, I think these clinical views help tie things together for you. Pancreas, know what comes from the pancreas. Know that the pancreas releases pancreatic juices, which are full of bicarbonate, which is alkaline. And that's important because the chyme that's coming from stomach is acidic. So this is important for neutralization. And there are also enzymes there that are going to digest various things. Okay, so that's pancreas. It has three main parts. It has the head, body, and the tail of the pancreas there. So that's that about pancreas. But no, I mean, we'll look at, actually, when you look at the nutrient digestion, you'll have, and that's table 26.2. You'll be able to look at like what enzymes are coming from there and what they digest. Table 26.1 summarizes the three main hormones. Motilin is not in this table for some reason, but these three are. So make sure that you know what they do. Cholecystokinin and secretin, their effect is the same. Their stimulus is different. So be aware of what the difference between the two is. And again, watch that Nearpod if you haven't yet. And I think I talked about that on Monday, maybe. Very briefly. And that's that. The large intestine structures, know the structures. So what is what? Labeling is always your first step to knowing, how everything works together. You have to know what is what first before you can learn how these structures work together. A little bit about histology. We do not have those plici here. There's no villi. You do have intestinal glands here, so that's kind of the difference between the histology here. It's not as wavy as what small intestine would look like. And we still have the columnar cells. There is a lot of goblet cells here, which we saw goblet cells in the small intestine also. And these are the cells that produce mucin, or that becomes mucus, and that serves for lubrication. But in the large intestine, you're going to have way more goblet cells, and this is because in the large intestine things get more and more solid, and so you need more lubrication in that area. So you would see more numerous goblet cells there. Motility and regulation of large intestine, be able to tell me what is haustro churning and what is mass movement. So haustro churning would be kind of similar to the mixing or segmentation in the small intestine, where it's just kind of mixing or being churned in the same place within the haustrum. And then mass movement is a movement of contents along the large intestine. Two reflexes that are associated there with defecation are gastrocolic reflex and the defecation reflex. Gastrocolic reflex is initiated when the food enters your stomach, and it causes contents to move from the small intestine into the large intestine. And so it causes mass movement, and it also causes mass movement. And then defecation reflex is initiated when the contents of your GI tract reach the rectum. So filling of the rectum stimulates the defecation reflex. There are two sphincters that regulate the exit of feces out of your body, internal and external. Internal is involuntary, external is voluntarily controlled. And so that's what kids learn to control whenever they're getting potty trained, is how to control that external sphincter. So that's that. For the nutrients and their digestion, what I want you to know from this is, where does the digestion of each nutrient happen, and what hormones, what enzymes are responsible for digestion of that particular nutrient. Okay, so know what enzymes are responsible for carbohydrate digestion, and you can look in your table 26 too, and it's all summarized there for you. You look at carbohydrates and you can see, okay, amylase, salivary amylase, pancreatic amylase, and then you have the maltase, lactase, sucrase, those. Dextrinase also. So carbohydrates have, I think, the most. Well, protein have a lot too, but carbohydrates do have a bunch. And and then know like where you would see each of these hormone or enzymes. I don't know why I want to call them hormones. They're enzymes. Know where you would see them active, like what part of your body would they be acting on. Proteins are easy to remember, I think, because if you remember just one enzyme that digests proteins, and that's pepsin, that's the first one that we talk about, then the rest of the protein digesting enzymes are going to either have the pep part in their name, or they're going to end with sin. Okay, so like if you look at like trypsin, ends with sin. You have carboxypeptidase. It has almost the whole word peptide in there. Okay, carboxypeptidate, it has peptid in there. So if you know that proteins are polypeptides, then you should be good. And you should know that these go with proteins, but also try to remember where they are active. Lipids, I think, might be the easiest, because all of the lipid digesting enzymes are lipases. So you have lingual lipase that comes from your oral cavity, but it's not active in the oral cavity. It's activated in the stomach. Then you have gastric lipase, and then you have pancreatic lipase. Okay, lipase for lipid, so that should hopefully be easy to match with lipids. And then we have nucleic acids. So there's the ribonuclease and deoxyribonuclease, that those should be easy to remember that they go with nucleic acids. And then you have phosphatase, you have nucleosidase also that are going to help with this, with digestion of nucleic acids. Nucleic acids are not like your essential nutrients. Nucleic acids, you can make, your body makes nucleic acids, okay? So, but carbohydrates, proteins, and lipids, you do have to ingest. And that's kind of going into chapter 27. When you look at the lipid break digestion, I would like you to pay attention, like special attention to that, to the lipid digestion, because this is a little different than carbohydrates and proteins. So for carbohydrates and proteins, I just want you to be aware, like the enzymes and where that happens, but you don't have to tell me like what the enzyme actually chops up, okay? I forgot to mention that. You do not have to tell me that maltose is broken down by maltase into glucose and glucose, okay? I don't need the outcome. Just know that they digest the carbohydrates. And same thing with the lipase. You don't have to tell me the chemical like bonds that are being broken. But I do want you to know for lipids that bile salts are involved. I want you to know that bile salts do not chemically digest lipids. The bile salts are going to surround, as they're being mixed with lipid, they're going to surround smaller droplets of lipid, and they're going to keep them from sticking back together. So imagine what would happen if you fill the water bottle with partially with water and then partially with oil. What would happen is they would separate. You can shake the water bottle, which is what would be happening like in your intestine, right? You're shaking this lipid and you're shaking the water contents of your GI tract. But if you stop shaking it, they will separate. The lipid will stick together, the water will stick together. And so as this is being mixed, these bile salts will surround the small droplets, and that will prevent them from sticking into larger droplets. This makes it easier for enzymes to come in and chemically digest those lipids inside of these, what we now call micelles. Okay, this is called emulsification. So the process of bile salts keeping the lipids from sticking together and basically emulsifying it within the water-based content, it's called emulsification. So if you've ever made like homemade mayo, you have egg and then you add oil to it. If you just like, and there's a process into it. I actually was never successful. It was just a big mess. But that's emulsification. That oil and that egg is basically not sticking together. Yeah. Now, if you added bile salts into the lava lamp, then you wouldn't have those nice big droplets floating around. Okay. So it would look cloudy. So imagine, don't try that, but imagine if you were to add, if that was lipid and water, kind of like in a lava lamp, if you added bile salts to it, it would get cloudy and the lipid would look like it's mixed in with the water. Okay. And then, nucleic acid digestion, just know which enzymes go with it. Nucleic acids are only digested in the small intestine. Chapter 27 is fairly short. We are skipping most of cellular respiration. So I want you to know the difference between essential and non-essential things. Essential versus non-essential. Essential have to be consumed or ingested or taken in a diet. And the non-essential, you can eat, you can get them as part of your diet, but your body is also able to make those, manufacture those. And we can also talk about essential versus non-essential amino acids. And this includes so essential amino acids are the ones that you have to get from diet. Non-essential, your body can make on its own. No, there's a lot of vocabulary here. A lot of vocabulary is a repeat from AMP1. So, and then there are also parts of this that are kind of an easier reading than the more scientific parts of the other chapters. Like, if you're reading about recommended daily allowance, you've heard that already before, and it's a kind of a common term. So you are already kind of familiar with that. Know the types of lipids or types of triglycerides and their fatty acids. There's a little bit of cholesterol. There's a little more about cholesterol a little later on in the chapter. Know what the deal is with the proteins. What are the complete proteins? What are the incomplete proteins? Most of animal protein is complete protein, and this is containing all of the essential amino acids. Incomplete proteins are going to have fewer amino acids. They don't have all the essential ones. And so people that don't consume animal protein might be at risk of being deficient in an amino acid. So they really have to pay attention to what kind of foods they're ingesting and are they getting all of the essential amino acids that their body needs. For nitrogen balance, know the difference between, when are you in a positive nitrogen balance versus negative nitrogen balance. The positive is when you are where you are ingesting more nitrogen balance than you're eliminating, and negative would be the opposite. Vitamins, so the macronutrients versus micronutrients, that will be part of your vocabulary there, but the macronutrients are the ones that you have to ingest a lot of, so your carbohydrates, proteins, and lipids. Micronutrients are your vitamins and minerals. And I do want you to know which vitamins are water soluble and which ones are lipid soluble. So water soluble are all the B's and C. And then you have A, D, E, and K, and these are your lipid soluble vitamins. The water soluble vitamins you can eliminate in your urine. The lipid soluble vitamins you cannot. And so if you have excess of these lipid soluble vitamins, then they can accumulate in your body and become toxic, but that would have to be like a chronic overload of these vitamins. And know kind of like generally what those lipid soluble vitamins are good for. And you can infer that if you look at the deficiency symptoms. So for vitamin A, since the deficiency can result in night blindness and child blindness, then vitamin A is obviously important for vision. Vitamin D, this one is important for proper calcium absorption, actually. If you don't have it, then your body is not absorbing calcium properly, and you might have bone problems. E, you can kind of skip E. And K we've already mentioned. K is important for blood clotting. Okay, so it's important in blood clotting pathway. You can divide vitamins into essential and non-essential as well. Essential are the ones that are going to need to be consumed, and non-essential are made in your body, and this include NADH and FADH2, which are part of the cellular respiration, part of the Krebs cycle there. So we don't really think about them as vitamins because our body makes it, so we don't really mention them as vitamins, but they are actually vitamins. Minerals and vitamins are organic molecules. Minerals are not. Minerals are inorganic ions. It's just a single element in an ionic form. And so for these, I do want you to know which ones are major, which ones are trace. Major ones are, you have to consume more than 100, I think more than 100 milligrams. And the trace are less, although I don't know why copper is in there in the trace where it says 900. So that's, some of these are unknown, but yeah, these are a lot of these are metals actually. Some of them are non-metals, but know which ones are major, which ones are trace. One interesting one is the iron is actually not a major mineral. Iron is a trace mineral, and that's because your body is so efficient in recycling it. However, iron actually happens to be one of the most common electrolyte or most common deficiency, mineral deficiency. So that's that. Guidelines for adequate nutrition. Read that, be able to read the label, know what my plate is. This is an easy reading there for you. So every time you sit down to eat a meal, it should be portioned like it is on that plate. So half of your plate should be fruits and vegetables. Are you doing that? You should. I should. I know I'm not doing that. I'm bad. But that's what they recommend for a healthy diet. Half of your plate should be fruits and veggies. And then the last part of 27 is regulating the blood levels of nutrients. So know how insulin and glucagon affect your nutrient levels in the blood, which that should be a little bit of a review from chapter 17, and know what absorptive and post-absorptive state is and which hormone is active when. Okay, so your absorptive state is when you eat and when you eat and digest. Okay, that's your absorptive state, and this is when your insulin is being active because insulin is going to lower the nutrients in your blood. And then that lasts about 4 hours, and then post-absorptive state kicks in, and this is when you're not eating, digesting, or absorbing. And this is when glucagon is important because it will go and release the nutrients that you stored into the blood. Cholesterol synthesis, you don't really have to go into the chemistry of cholesterol synthesis. What I do want you to know about cholesterol is the difference between VLDLs, LDLs, and HDLs. So the ones that are low, either the very low or low density lipoprotein, these are going to carry the lipids to tissues. And they also have a bigger storage capacity. Your HDLs are high density lipoproteins. These are going to have a lower storage capacity because they are composed of more protein. So the space compartment is smaller. And these will take lipid from the tissues. So when you're looking at someone's cholesterol, what's important is to, rather than looking at individual numbers, it's important to look at the ratio of these. So, if you have very high VLDLs or LDLs, well, if you also have high HDLs, they should be able to balance it out. But if you have too many of the low ones that are taking lipid to the tissues and not enough HDLs, then sometimes these low and very low density lipoproteins will deposit lipid where it's not supposed to be deposited. And if you don't have enough HDLs, they don't have time to clean it up. And that can lead to clogging up of your arteries and things like that. And then 27.7, central role of respiration. You don't need to go into the steps. What I want you to know that you can add any nutrient, like any of those three, proteins, lipids, or carbohydrates, into the process of cellular respiration here. Whatever nutrient you begin with, you're going to break that nutrient into acetyl-CoA. So notice that on that table, everything, like whether you start with glucose or with the lipid or with the protein, it leads to acetyl-CoA. And these are reversible reactions. So you can actually go backwards and you can interconvert between nutrients. So you can begin with protein and it goes into acetyl-CoA, and then you can convert it into glucose, okay, or into lipid. If you have diet that's really high in protein, that protein could be actually converted into lipid eventually if you don't need all that energy. So nutrients are interconvertible, but you don't need to do any of the electron transport chain and citric cycle and all that, okay? And then energy and heat, the last part, vocabulary from this. Know what is metabolic rate, what is basal metabolic rate, total metabolic rate, how is it measured, what are the things that affect your metabolic rate, be able to discuss that. And then temperature control, there's nervous system control of temperature. And we talk about temperature here because it has to do with the rate of metabolism. So the higher your metabolic rate is, the more heat your body is producing as a byproduct. So your body can regulate the heat or temperature by nervous system, where the hypothalamus is going to receive stimuli and then do certain things, depending on, okay, is the body too cold or is it too warm? And then there's certain mechanisms that happen here. And then hormonal control, I want you to know that the most important hormone that affects your temperature is the thyroid hormone. But there are some other ones like epinephrine and norepinephrine also do affect your temperature. Testosterone also, higher levels of testosterone are going to increase temperature. So that's why it seems like males are always like more hot than females. I don't know. My husband is always warm in our house and I'm freezing and I need like 20 blankets. So, so that would be a reason. Yeah. Right. Except for my mother-in-law's house, but she's after menopause. So maybe something is off with her hormones. I don't know. And my father-in-law is always cold. But yeah. So that's it. That's that for 27. That was a very quick overview of 3 chapters.
How's it going to be broken down? Is it all kind of 30%?
I wouldn't say there's 30% from the nutrition, just because that chapter is much shorter.
Thank you.
So if I were to guess, I don't know. If I were to guess, it would be maybe like 40, 40, 20.
Okay. And then which histology files should we know?
What do we have? We have lung, trachea, and then whatever is there for digestive system. I think there's small intestine, large intestine, maybe pancreas. Pancreas was also a part of
pancreas is there.
Yeah, okay. But yeah, for respiratory system, it's just lung and the trachea. And for digestive system, I don't know if I have liver in there.
There's trachea, stomach, esophagus junction, small intestine with the Peyer's patches, the liver. So all those?
Yeah, what's after liver? I'm not looking at the file right now.
Kidney. So I'm guessing.
Yeah, no, so stop there. So kidney is going to be next chapter.
Okay.
All right.
And anything to do with anatomy and labeling that we should focus on? I know you mentioned the teeth.
Yeah. Yeah, no, the teeth, but I don't think anything is like, I don't know. Definitely know the first, the first few figures of each chapter, because that gives you like the major stuff. The one that's tricky is the respiratory system one, where you have the alveolar sac, the one that I mentioned earlier. And then another one is with the ducts in the digestive system, chapter 26. To pay special attention to those, just because those are, a lot of students do make mistakes on those too.
And then process labeling for, I'm guessing the respiration for pulmonary respiration.
Yeah, so be able to like arrange the steps of the respiration. Is that what you meant?
Yeah, that's exactly what I meant.
Yeah, anything else? And I mean, you have, it's only Wednesday, so you still have a few days. So if you think of anything, as you're reading something or something doesn't make sense, let me know. And good luck. And then we don't meet next week. It's Thanksgiving week, right? But I didn't want to like push this exam until after Thanksgiving because you would forget. You need to be able to name them. I don't think I'm going to ask you the numbers though. But you do need to know numbers of teeth total. So no number, like how many deciduous you have and how many permanent teeth you have. You do need to know that. But I don't think I'm going to ask you like specifically how many incisors or how many molars you have.
I don't know if it'll have it this time, but I don't think anyone needs it anymore, but the recording reminder.
You know what? It is there and it's before you click begin. I actually checked it and several people are like, I didn't see the reminder. But then when I watched the videos, it's there. Yeah, so it's like when you first click, no, it's like you go into your Connect, then you click on, I think you click on begin, and then it tells you like you will have this much amount of time. There's a paragraph above that. And that's the only way I can put it in there. So that's where it is. Okay. I think so. Yeah, exam 4. I wish there was like a better way to make it obvious for you guys because yeah, there were several people that said it wasn't there. And then when I checked on the video, there it was. So it's not, what that tells me is not very visible and in your face. And that's the only way that I can do that. So, okay, well, I mean, we are making pretty good time here, hour and 7 minutes. So if you think of any other questions, let me know. You have a great evening and great Thanksgiving. Stay safe and good luck on the exam.